All right, so we're going to start our final chapter for the semester, final chapter for 2402, which is the reproductive system. So let's start by looking at what males and females have in common with their reproductive systems. They both have primary reproductive organs that are called gonads. So if a question asks you what the primary reproductive organ is in a male or a female, it is the gonad. Now, it's the gonads are different in males and females. So for females, the gonads are the ovaries, and in males, the gonads are the testes. And the gonads are what are responsible for producing what's called the gametes. The gametes are your sex cells. So sperm for males and oocytes for females. Keep in mind, oocytes can also be called ova sometimes, but it's basically just the egg. Now, the sperm and the egg is what's going to unite at fertilization to start the formation of a new individual. The gonads are also responsible for producing a large amount of sex hormones. Primarily, it's going to be testosterone in the male, estrogen and progesterone in the female. Now, yes, females have testosterone and males have estrogen and progesterone, but just in much different quantities. So the testes are responsible for producing sperm and a large amounts of testosterone. The ovaries are responsible for producing the oocytes and large amounts of estrogen and progesterone. Those sex hormones are responsible for maturation, development, and changes in the reproductive system. Okay, so both sexes also exhibit accessory reproductive organs, and that can be ducts that carry the gametes away from the gonads towards the site of fertilization. So that would be in females. So it's going to carry the oocyte away from the ovary towards the uterine tube where fertilization would take place. In males, it carries it from the gonad all the way outside of the body. Both male and female reproductive systems are primarily non-functional and dormant until a time during adolescence known as puberty. At the onset of puberty, the external sexual characteristics start to become more prominent, and that's when the reproductive organs are going to become fully functional in both males and females. So here's kind of a little rundown of what actually starts the process. Of course, it's going to start by an action at our master endocrine regulator, the hypothalamus. So the hypothalamus starts to release gonadotropin-releasing hormone, or GnRH. GnRH is going to act on specific endocrine cells in the anterior pituitary and cause them to release follicle-stimulating hormone, or FSH, and luteinizing hormone, or LH. So prior to puberty, FSH and LH are basically non-existent. While our anterior pituitary has the capability of making them, it just never does because that GnRH has never been released from the hypothalamus, and that's the stimulus that the anterior pituitary needs to make it. So as the levels of the FSH and the LH continue to rise, the gonads are going to respond to those hormones. And the gonads respond by producing significant levels of sex hormones. And then those sex hormones are what's responsible for starting the process of gamete and sexual maturation. So simplified version, you've got gonadotropin-releasing hormone, or GnRH, from the hypothalamus. That leads to FSH and LH release from the anterior pituitary, and then that is going to start the process of hormone release from the gonads, which causes gamete maturation. So looking at the gametes, females typically produce and release a single gamete called an oocyte or an egg or an ovum monthly, while males produce a large number, about 100 million per day, of their gamete, which is the sperm cell, daily. So about 100 million sperm per day being produced. The sperm are going to be stored for approximately 1 day, and then if they don't end up getting expelled from the body at that time, they get reabsorbed. So they kind of have a limited time frame in which they're good. In the male gonads, we're just really churning out a ton of sperm. Leave them there. Hopefully they get a chance to do their job, and then if they don't, it's too bad, so sad, they get reabsorbed, and then we'll just make new ones. Humans pass on their traits to their offspring, And the hereditary information is carried on our chromosomes, which we know is made of DNA. Somatic cells, or body cells, contain 23 pairs of chromosomes. 22 of those are referred to as autosomes, and the last pair is referred to as sex chromosomes. So since we have 23 pairs, it's a total of 46 chromosomes per cell. So I use the term somatic cell. Basically, your somatic or your body cells refer to any cell or every cell in your body with the exception of the sperm and the egg. Okay, so every cell in our body, with the exception of sperm and the egg cells, have 23 pairs of chromosomes, so 46 total. 22 of those are referred to as autosomes, and 1 is a pair of sex chromosomes. So the autosomes are chromosomes that contain genes that code for a variety of cellular functions, most of our human characteristics. So your eye color, your hair color, your height, skin pigmentation, all of those things are going to be carried out on your autosomes. A matching pair of autosomes are called homologous chromosomes. And I'll kind of go into that a little bit in a minute as well. Then you've got the pair of sex chromosomes. Pair of sex chromosomes either contain 2 X chromosomes or 1 X and 1 Y. Those are the chromosomes that are going to determine the biological sex of an individual, whether somebody is a female, in which case it would be XX, or male, in which case it would be XY. So your autosomes are going to code for most of your characteristics and features. Your sex chromosomes are basically what determines whether you're male or female. Okay, terminology. Hopefully you guys are familiar with this kind of after running through the lab activity. A cell that contains 23 pairs of chromosomes is said to be diploid. So if something has 46 total chromosomes, it is a diploid cell. It's said to have 2 N chromosomes, where N is the number of unpaired chromosomes. So N in a human would be 23. So 2 times 23 would be 46. Okay, so that's a diploid cell. If the gametes were diploid, then each offspring would receive 2 sets of paired chromosomes, or 4N. So let's say if the egg had 46 chromosomes and the sperm had 46 chromosomes, then when those met at fertilization, the new individual would have 92 chromosomes. And it would continue to grow, with every generation. And that's just simply not compatible with life. We have to have 46 chromosomes in an individual. So the gametes from either sex, whether we're talking about a sperm or an egg, those are referred to as haploid because they only have 23 chromosomes, not 23 pairs. So haploid individuals have what's referred to as 1N, just 23, so just the N. In this way, when the oocyte, which has 23 chromosomes, fuses with the sperm, which has 23 chromosomes, it's going to result in an individual or a cell that has 46 chromosomes total. Okay, so all of your other cells have 23 pairs of 46 chromosomes. Those are diploid. The gametes, so the egg and the sperm, they can only have half the amount because they have to fuse together to create a new 46 chromosome individual. So those are haploid, or just N. They contain 23 chromosomes only, not 23 pairs. Okay, so in order for the gametes to become haploid, they have to undergo a special type of cell division called meiosis. So meiosis is a type of cell division that is going to take place only in the sex cells. You're only going to get meiosis happening in the oocytes and the sperm. And it starts out with a diploid parent cell, but in the end, you're going to end up with haploid daughter cells that are called gametes. So you're going to start with a diploid cell, you're going to go through the process of meiosis, and you're going to end up with your haploid gametes, which are going to be your oocytes and your sperm. So down here, we're contrasting some of the primary differences between mitosis and meiosis. Because we remember mitosis, right? Mitosis is the process that most of our body cells, all of the cells in our body except sperm and eggs, use to replicate. That's mitosis. Meiosis is the process that we use for our sperm and our eggs. Okay. So mitosis produces two daughter cells that are genetically identical to the parent cell. So during mitosis, basically you have a single cell, everything in the cell replicates, and then it splits in two. So we end up with two cells that are genetically identical to the parent cell. In meiosis, you get four daughter cells that are genetically different from the parent. At the end of meiosis, you've got a new mixture combination of DNA that's unique from the cell that you started with. And you've got four of them instead of two. Okay, other differences. Mitosis produces daughter cells that are diploid. Meiosis produces daughter cells that are haploid because we're making those eggs and the sperm. Also unique is meiosis includes a process called crossing over, and we'll look at that. But crossing over is where genetic material gets exchanged between something called homologous chromosomes. And when that happens, we actually create a new chromosome that has never existed before. It's a unique chromosome. And that only happens in meiosis. The cell phase that happens just before meiosis is called interphase. Now, during interphase, the DNA in each chromosome is going to be replicated, where it's duplicated exactly in the parent cell. The replicated chromosome is composed of two identical structures called sister chromatids. So that's what you're seeing here. This is one chromosome, but it's made up of two sister chromatids. So this is just a replicated chromosome, basically. Each sister chromatid contains an identical copy of DNA, and then the sister chromatids are attached at the central region called the centromere. So this is a really good slide to visualize a couple of things. This is a single chromosome. Okay, so a chromosome consisting of a single chromatid and a centromere. So this is one chromosome, right? When this chromosome replicates, as it's going to do before a division, this is what it looks like. So this is also one chromosome. So don't let that confuse you. This is not two chromosomes. This is one chromosome, but it's a replicated chromosome. That's why it has the two sticks, and each stick is known as a sister chromatid. This is a homologous pair of chromosomes. So here's one chromosome, and here's another chromosome, but they're a homologous pair. What does that mean? When I say we have 23 pairs of chromosomes, we literally have chromosome 1, chromosome 2, chromosome 3. They each code for basically the same thing in all individuals, but we have different variations of genes, which is why we don't all look the same. So let's say, just as an oversimplification, let's say chromosome 1 codes for hair color, chromosome 2 codes for eye color. That would be the same across all individuals. We just have different hair color and eye color because we've got a variety. We have allele variation, which I won't go into the details of that. But the reason we have 23 pairs is because you get one of those chromosome 1s from your mom and one from your dad. You get a chromosome 2 from your mom and a chromosome 2 for your dad. So this would be like the chromosome 1 that you got from your mom and the chromosome 1 that you got from your dad. So it's referred to as a homologous pair. Okay? During meiosis, the homologous pairs of chromosomes are going to be separated whenever the cell divides. And the result is going to be that you have two cells, and in each cell you're going to have 23 chromosomes instead of 23 pairs. And those chromosomes, so it would be haploid at that point because you only have 23 chromosomes. The problem is those chromosomes that you end up with at the end of the first stage of meiosis, they're still replicated chromosomes. So that's not the genetic composition that we need at that time. We need 23 non-replicated chromosomes in order to be genetically compatible to be the gamete. Okay, so meiosis has two different phases, meiosis 1 and meiosis 2. And each one of those phases is broken down into 4 different phases or subphases. and it's prophase, metaphase, anaphase, and telophase. So it looks just like mitosis in terms of the different phases. For meiosis, we put a number 1 or a number 2 after it, depending on if we're in meiosis 1 or meiosis 2. So meiosis 1 is made up of prophase 1, metaphase 1, anaphase 1, and telophase 1, and then cytokinesis. And then from there, these will move on to meiosis 2. Let's look at prophase 1. You have homologous replicated chromosomes in the parent cells that form what's called a tetrad. This is sometimes called a bivalent instead of a tetrad. Keep in mind here that the chromosome here consists of 2 sister chromatids because it's replicated. The process by which homologous pairs line up is called synapsis. Okay, so in this individual, so we know in a human cell, there's 23 pairs. So they don't want to draw 23 pairs. That would be too much. So to keep it simple, they're drawing 3 pairs. So we could call this chromosome 1. So this is chromosome 1 from mom, chromosome 1 from dad, chromosome 2 from dad, chromosome 2 from mom, chromosome 3 from mom, chromosome 3 for dad. So they're leaving it at 3 to keep it simple. So the homologous pairs line up. So both of the chromosome 1s come together. Both of the 2s come together. Both of the 3s come together. This happens for all of our chromosomes. And again, that's called a tetrad when they come together, also known as a bivalent. So as the maternal and the paternal chromosomes come super close together, something that happens is called crossing over, where the homologous chromosomes within each tetrad get a little bit of an exchange of genetic information at that time. So you can see this was a solid red chromosome and this was solid blue, but where it got really close together, a little bit of that blue jumped over onto the red and a little bit of the red jumped over onto the blue. Okay, so that's crossing over when that happens. A portion of the genetic material in the sister chromatids on the maternal chromosome gets exchanged with the same portion of genetic material in the chromatid on the paternal chromosome. That's what I just said. This is a way to ensure genetic diversity, because once that swap of genetic material happens, this is a new chromosome that has never existed in the history of time. So we continue to create new variety and new variation by using this crossing over technique, and that is very compatible with the continuation of life. We need that new variety happening in our genetic material. Okay, note that after crossing over, the sister chromatids and the replicated chromosome are no longer identical. Okay, so this chromatid looks different than this chromatid because this one got a little bit of the blue, but this one didn't. So after crossing over, the sister chromatids are no longer identical where they were before that. Okay, prophase 1 ends with the breakdown of the nuclear envelope. So all of our chromosomes are contained in our nuclear envelope, but we can't separate them into separate cells if they're contained in that envelope. So in prophase 1, your homologous pairs come together. That's a process called synapsis. Crossing over happens. We talked about that. And then it ends with the breakdown of that nuclear envelope, so we can let the chromosomes out because they're going to need to eventually separate. Next thing that happens is metaphase 1, where the homologous pairs of each tetrad line up on either side of the midline or the equator of the cell. So metaphase is always, to me, the easiest one to remember because it's where they're all kind of lined up. So here's kind of the midline of the cell, and you've got the pairs kind of on either side of that, right? The alignment of the paired chromosome is random with respect to whether the maternal or paternal chromosome of one pair is on either side of the equator. So you'll notice that dad's chromosome is on the left and mom's is on the right for this one. But over here, mom's is on the left and dad's is on the right. And so it lines up completely randomly. And this is called independent assortment. And this is also very key to ensuring genetic variability. because what's basically going to happen is everything on this side is going to end up in one cell, and everything in this side is going to end up on a different cell. And you can imagine, let's say this were to split right like this, and all of these went into one cell. Okay, that's, that's one genetic variety. It's got this genetic makeup. But if this were to end up on this side, if this red and blue were to switch, then you would have an entirely different genetic makeup. So depending on which chromosomes line up on which side of the midline of the cell, for the cell to then later divide, it's going to change the genetic makeup of the subsequent cells. Okay? So that's called independent assortment, where it just, it's kind of this random alignment of which chromosomes are on which side of the equator. And independent assortment is, in addition to crossing over, is one of the main things that allows for genetic variation. Okay, so spindle fibers start to form by the microtubules extending from the centrioles. So here's our centrioles, and you get these spindle fibers forming. So they're going to reach out towards the center of the cell, and they're going to attach to the centromere of each homologous replicated chromosome. So right here, and right here, and then also on this side. So you're getting the attachment of the spindle fibers to the centromeres. Okay, in anaphase 1, those spindle fibers are going to start to shorten, and that's where you start to get the pulling or the dragging of the chromosomes to either side of the cell. So at this point, the homologous pairs of chromosomes are starting to get separated, and again, pulled. These are going to get pulled over here. These are going to get pulled over here. So a maternal chromosome consisting of two sister chromatids is going to get pulled to one side, while the homologous paternal chromosome is getting pulled to the opposite side. So this is your maternal chromosome getting pulled over here. This is your paternal chromosome getting pulled over here. Because of this process, each potential daughter cell receives only half of the starting material, so only half of the chromosomes. So by the time this process is over, it will have 23 chromosomes instead of 23 pairs of chromosomes. Again, those chromosomes are still replicated, so we can't consider it a gamete until we separate the sister chromatids. Okay, so in meiosis 1, we're separating homologous pairs. In meiosis 2, we're separating sister chromatids. Telophase and cytokinesis. So the replicated chromosomes arrive at opposite ends of the pole, and then the nuclear membrane starts to reform. At this point, we can reform the nuclear envelope and start to kind of package them back up there. Then you've got your cleavage furrow. That's this indentation right here. That starts the process of dividing the cytoplasm. Okay, and now again, each daughter cell has 3 replicated chromosomes. Here's 3 replicated chromosomes. Here's 3 replicated chromosomes. In a human, of course, that's 23. It's just in our diagram, it's 3. So in a human, it would have 23 replicated chromosomes per each daughter cell. The 2 cells that are formed at this stage have to undergo that separate round of division, which is going to be meiosis 2. And again, the purpose of that is going to be to separate the sister chromatids, because we don't want replicated chromosomes for our final product. Okay, again, after meiosis 1, the 2 daughter cells formed each contain 23 replicated chromosomes, each consisting of 2 connected sister chromatids. And meiosis 2, those chromatids are going to be separated and become single chromosomes. So if this is one of the daughter cells from meiosis 1, and this is the other daughter cell from meiosis 1, let's just follow the top pathway. They're basically going to line back up again. This would be a metaphase. This would be an anaphase. We're going to separate them. And then this would be the final product, kind of. It's almost final if we can cleave that, where you can see the product is 1, 2, 3. So 3 single chromatids. Again, in humans, that would be 23 single chromosomes. Okay? But one of the cells from meiosis 1 produces 2 final cells. Here's your other one, 2 final cells. So we end up with 4 cells total at the end of meiosis 2. Okay, prophase 2. The second prophase event of meiosis looks a lot like the first prophase in the new cells, where the nuclear envelope is breaking down to allow access to those chromosomes. Major key important point, crossing over does not occur in this phase because you don't have homologous chromosomes. We don't have homologous pairs in prophase 2 like we did in prophase 1. So in prophase 1 you had your maternal and your paternal chromosome, and that's what allowed for crossing over. But we separated those in meiosis 1. So in this one you only have 1 chromosome 1 and 1 chromosome 2 and 1 chromosome 3. And we don't get crossing over between different chromosomes. So it doesn't have a homologous pair, so crossing over does not occur in prophase 2. Okay? Metaphase looks like metaphase always looks, like it looks in mitosis, like it looks in meiosis 1. Everything lines up in the middle of the cell. Okay, and you start to get spindle fibers attaching to the central region of each chromosome. In phase 2, those spindle fibers are going to shorten, and we're going to separate the sister chromatids, and we're dragging the sister chromatids to either side of the cell. Telophase 2 in cytokinesis, so the single chromosomes have arrived at opposite ends of the cell. The nuclear envelope starts to reform, and we start to get the division of the cytoplasm. So you get that cleavage furrow. This will eventually come all the way in until you separate the cells into 2 different cells. Okay, these meiotic processes produce 4 daughter cells from 1 original single cell. The daughter cells are haploid because they contain only 23 chromosomes and not 23 pairs. 22 of those are autosomes, and then 1 is your sex chromosome, so either an X or a Y. So just like all the other clinical views, I'm not going to kind of read through the whole thing, but I do want you guys to do that. I'm going to actually see if I can find a video that shows non-disjunction. Basically what non-disjunction is when you get an inappropriate separation of the chromosomes or the chromatids. So you can see, if you look at this one, and I don't know why there's this white whatever this is in there, but this would be separation of the chromatids. So you've got a chromatid moving over here and a chromatid moving over here. If that doesn't happen properly and the entire replicated chromosome gets pulled to this side, what that's going to do is it means for the 2 daughter cells that get produced, this one got an extra chromosome. So because the chromatids didn't separate. So where this cell should have 1 red and 1 blue, it has 1 red and 2 blues. That's called a trisomy, because where you should only have 2 copies, 1 maternal and 1 paternal, if you have 3 copies due to non-disjunction, it's called trisomy. And what that's also going to do is it's going to lead the other cell that should have gotten one of these chromatids, it didn't get any of them. So this should have a red and a blue, but it only has a red. So this one in a human would only have 22 chromosomes instead of 23. This is called a monosomy because you only have 1 instead of the 2. So most chromosomal abnormalities are what we call embryonic lethal. They don't even continue to develop to become a full fetus. There are a few that are compatible with life. Trisomy 21 is probably the most common one that we're all familiar with. That's Down syndrome. So that's trisomy of the 21st chromosome, where you get 3 copies of the 21st chromosome. So again, I'll try to show a video, I'll try to find a video that actually shows this, but kind of read through this and be familiar with the information in here.